Hello guys, welcome to this channel. I know this that you have so many confusion in this part of activity because I personally had so many confusion. Let's get into this video and let's dive in this activity that how can we clarify all this confusion. So let's start. Basically we have this activity in our NCERT book, which says first that we need to take two test tubes which are filled with water, and precisely it has written that we need to take 10 milliliter or 15, okay, 10 milliliter of water, and then we have to add drop of oil into it. In both the test tube we have to add drop of oil into it, and one more test tube we need to have in which we have you know soap solution. We have soap and we have pour some water into it. So this is basically soap solution that is there. Now we need to pour soap solution in one of the test tube. Let's suppose I pour this soap solution into this test tube, and for example this is now oil plus water plus soap solution. So basically what will happen, and what will happen in this test tube where we have oil plus water. Now we need to shake both of them for equal amount. We need to shake them, and what will happen afterwards, by shaking we need to observe that what will happen. In NCERT we have been given this line that can you see the oil and water layer separately in both the test tubes immediately after you stop shaking them. Okay, so basically after shaking we will notice that we cannot see any layer in both the test tubes. Why I'm saying this, I have a point. Basically in both the test tube the drops of oil and water will get mixed to each other. In both the test tube this will happen, and after some time when we read the next line which is there, leave the test tube undisturbed for some time and observe. So what we'll observe after keeping these two test tube for some time, we'll observe that in this test tube we would able to see the separate out layers. Basically what will happen is the layer of oil and water will separate out, but in this test tube the layers of oil and water will not be separated out, or basically will not be able to see those layers of oil and water. In this case there is no layer separation. Okay, but in that a part, you know in a test tube we have layer separation. Why this happen and how this happens, everybody has their own explanation. Some says that in this test tube oil get dissolved in water, but this, trust me on this is absolutely wrong. This is absolutely wrong, and I need to highlight this point that guys, you are not able to understand this part because teachers tell you wrong part, right? Wrong thing. So today I'm going to tell you or teach you how you need to see. You can make your own answer as well, but the concept, the logic has to be the same. So you need to understand that how you can take out the easily, how you can easily take out concept from this, right? So basically what is concept, let me teach you, then you can easily frame your answer. Basically the concept is that emulsion, this solution which you are able to see where there is no layer separation, oil and water is not dissolving. Basically oil is not dissolving in water. What is happening is we are getting emulsion. The soap which is dissolved in this test tube B, let's suppose we name this test tube to be B. Now this oil and water will get dissolved. Why? Because oil is hydrophobic in nature. Okay, oil is hydrophobic in nature. It is not going to dissolve in water. This can never ever gonna happen. So basically oil is not gonna dissolve, and oil is basically non-polar. We all know that oil is non-polar and hydrophobic in nature. Now what will happen is, and one more thing that we know is soap is also non-polar and soap is also a hydro. Basically soap is non-polar plus it also has ionic part. So basically what it will form is, it will form, this is oil, and This is basically soap structure. You all know that. I don't have to tell you, but this is the soap structure where this is a long carbon chain, but here this part is basically ionic part, which means it has charges on it, plus minus charge, those charges it has. So this is ionic part, right? So basically it will form, this part is hydrophobic in nature. That's why it tries to separate out from water, but this part is hydrophilic in nature. So this part tries to be in water only. So we have the mixture of water, oil and soap. So what will happen, the oil part which is there, it will be surrounded by the hydrophobic part, because oil is also non-polar and hydrophobic in nature. So it will get attached to hydrophobic part which is there. So many hydrophobic part of the you know soap will get attached to this oil, and will have a spherical part over here. And the hydrophilic part which is there, water loving part we say, that is in the water only. So both are happy. Now this part is also happy, which is hydrophobic, because it formed a circular, spherical kind of structure. That structure would not allow water to enter inside. So the hydrophobic part is happy right now, and also the hydrophilic part is happy because it is surrounded by the water, which is, like for example, this is actually water loving part, so it wants water and it is surrounded by water, so it is also happy. And this particular is also happy, because we know that hydrophobic part is not getting water, as it is enclosed in that spherical, this is a spherical, you know the structure which is there. So we need to understand that this is not allowing water to enter inside. That's why we are not, they are not disturbed. Basically they are also happy. So what I'm trying to explain by this missile formation is around all the parts of oil. Basically when we shake the oil layer gets separated into so many small small parts. Now each part is actually surrounded by this missile, and that's what we call as emulsification. This is where emulsification concept comes, which I guess nobody uh, explains rightly. But let me tell you, this is also given in biology part. If you remember the first chapter in biology, which was of life process, and in that chapter, in digestion, human digestion part, we had emulsification of fat. You remember the pancreas secreted bile juice, and bile juice help in emulsification of fat. What was that emulsification? That was basically breaking down of, let me write it very nicely, breaking down of fat globule. Basically there were big fat globules, they need to be broken down into small so that further digestion process can take place. That's why we had that process called emulsification. Now in this emulsification we had the conversion of fat globule into small fat molecules. Now here also the same case happens. Basically we had this layer of oil. Now this layer of oil is getting, due to shaking, this layer of oil is getting separated into small part, and each part is surrounded by this missile, right? Each part is surrounded by this missile. And since each part is surrounded by this missile, I'm not able to draw it carefully, but you need to understand that each part is surrounded by the circular structure that water is not able to enter. So therefore this whole parts of oil is dispersed in the water. Let me show you with a figure so that you can easily understand. This is a beaker where we have oil plus water and plus soap. So soap is forming missile around the oil, right? Soap is forming missile around the oil. This is the oil particle which I'm drawing with the gray color, and this is oil particles, and this is basically the formation of missile around each of them, which are not allowing them to again form the layer. Can you understand the concept here? Basically we have the water, which is indicated by the blue color. As you can see, we have the water, and this soap missile, actually we can say, this missile is not allowing this soap, this missile not allowing this oil part to again form a layer, because this missile is not allowing the other oils to form or to join into them. There is one oil part, there is one oil part, they are not able to join together because of the missile formation which is there. So you need to understand that missile is the reason due to which oil is not able to join to oil. Basically, some part of oil is not able to not to join to some other part of oil, so they're not able to form layer. That's why. That's why here come the reason why we are not able to see the layer, why there is no layer separation, because we had missile around the portion of oil. When we just mixed it, let me show you, when we just shake it, then we were not able to see. We have shaked it, right? Then it converted into small parts, oil parts, and these oil parts were not able to join together. And why they were not able to join together? Because obviously we have the reason that there was missile formation. Now I hope you understand that what do you mean by emulsification. Shit, sorry. What do you mean by emulsification? And now I hope you also understood that what actually happens when you are not able to see the layer in both the test tubes. Even if you are said that in the second test tube, in the first test tube you were able to see the layer because there was no soap solution added into it, but in the second solution you were not able to see the layer because simply it was due to emulsification, which is the formation of micelle around each part of the oil, which are not allowing them to join together and not able to form a layer on the water. So hence this is the answer to this question, that why they were not able to see, we were not able to see the layer, we are not able to see the separate out layer. Okay, so this is the case, guys. Now I hope you understood each and every concept carefully and nicely. This is Aditi Singh, guys, signing off with this. Bye.